All right, let's just talk about taking a few tissue samples. Tissue samples are great as long as you have soil and paste data. Just taking a tissue sample is not the most informative thing because it can be very confusing. But in taking a tissue sample, there's two areas that we want to concentrate on sampling. Number one is the first full mature leaf. In this case, in the tomato plant, we could either take this one or this one. So I could, if I pull this one off, now you can, in this case with tomatoes, you either take the leaves or you strip the leaves off and take the petiole. In most cases, if you were looking at corn or beans or something, you'll take the whole leaf, the first mature leaf. And so that's not these leaves on top, it'll be down just a little bit below the top. Now, the other great way to analyze tissues, which is a better help if you're going to just do a tissue sample, then you can also look at sampling the lower leaves and then sample the higher leaves up on top. So what happens is nutrients will be translocated from the low leaves to the top. And those nutrients, and so if that's the case and we're young in the growth cycle, if these leaves are showing deficiency, like this tomato plant here is showing some potassium deficiency, is burning around the outside edge. That's a highly mobile nutrient, so it's going to move up into feeding these new plants and the tomato set. So the best way is to do both the upper leaves and lower leaves, but it's really helpful too for somebody like me if I'm going to look at tissue data to look at this as well as a paste test. And that's part of the soil test. The paste test shows what the plant is actually seeing. So by looking at that data and this data, that will match up very well. The standard soil test will not match up well with this, but the paste data will. So once you have the tissue data, whether it be tissue or the petioles or whatever, again, go and put them back in a paper bag. We don't want to put these into a plastic bag because they'll mold and by the time they get to the lab, they'll be a slimy mess. So put them in a paper bag. You can actually let them dry out. They can get crispy. The lab's going to dry them out artificially anyhow, and then grind them up and run the analysis. So the key thing is put them in a paper bag or just leave them set out to dry, and then you could put them in a plastic bag, but don't put them in raw.